we are going to speak about the blotting techniques. So we separate the DNA, RNA or proteins. The separated DNA, RNA or proteins has to be identified. So in our interest, so we have our interest DNA, RNA or protein that has to be identified. And that identifying technique, like extraction technique, we call it as a blotting. So we have basically in molecular biology, we use blotting techniques to separate to identify or visualize the DNA, RNA, or protein of our choice. So what is the principles behind this technique? First is a separation technique. So in the separation technique, we have electrophoresis. So during electrophoresis, we separate the DNA or RNA through agarose gel electrophoresis. For protein, we separate through the polyacrylate gel electrophoresis. So through gel electrophoresis, what happens? The DNA or RNA is separated based on their size. We know molecular markers are there, and we do have all this DNA separated bands formation. At the same time, RNA also separates through the same process. Now, once it is separated and the proteins are separated through the same polyacrylate gel electrophoresis. Now, this gel, the separated bands formed gel, is transferred to the membrane, and then we give them a supporting matrix. We kind of have a grid. We have a chamber here. We can see one of the chamber here. In this chamber, we have a gel followed by we have some spongy pads. We keep on the surface, we keep a membrane, the nitrocellulose membrane. There are some other membranes as well as upon that we keep some of spongy spongy supporting matrices. We also call it as a pads, and then we have a grid. So we cover this entire transport membrane gel in this, and then we keep it in the buffer, and we pass the current to it. So now what happens in the presence of the buffer, whatever the DNA is there, which is separated based on the size, is getting transferred to the membrane. The membrane is nitrocellulose membrane over here, or you can have another membranes also. There are a lot of other membranes are used. So the technique, the passing of the technique, we call it as a capillary force it is used. So that is what exactly happens. Once the transfer happens, let's say exactly whatever sample we have it in the gel, in the band form, same way it gets transferred to the membrane. Now we have nitrocellulose membrane, nylon membrane. So now what happens in the given bands of the DNA, RNA, or proteins, we have our protein or our DNA of interest. Now, how do we detect? We have a probe, radio labeled probes. That means our DNA of interest, we want that our probe to be annealing with that DNA of our interest, which has the radio label. So what happens now when we add our radio labeled probes, will anneal with the DNA of our interest, and then this annealed DNA of interest, what happens? We analyze under the X-ray. So whichever the DNA has been annealed, that gives the color. It gives the luminescence. So this is how we detect the DNA of our interest. So same way it goes for the proteins or the RNA. So basically, blotting technique is used from the gel, what we extracted from the gel, whatever the gel we use, the gel DNA to extract the DNA of interest. So this is what exactly the blotting is. So in the DNA blotting technique, we have three types. We call it as a southern blotting, northern blotting, and the western blotting. So southern blotting is used for the detect the specific DNA sequence. The name Southern came because of one of the scientists called E.M. Southern. So initially, the beginning, the Southern blotting was identified, discovered. So identified. So that technique we call it as a Southern blotting. Later, we found out Northern blotting. So Accordingly, the name Northern was kept. It was nothing to relate it to the scientists, but after Southern, they kept the Northern. And then the protein detecting technique was found out. Then we did the Western blotting. So that was named the Western blotting. So this is how exactly the technique names was found. So how exactly these techniques are done? So first one is a southern blotting. Under southern blotting, we have an agarose gel. So we have wells on that. So we put the molecular markers in one of one of sample loader, and then in other gels we put the DNA of our interest. So then we make this electrophoresis to occur. Once the electrophoresis is done, the DNA migrates, and according to the size, as we know, agarose gel electrophoresis, you might know. So according to the DNA gets separated size. Now, once it is gets separated, the bands are formed, and this gel now is transferred to the nitrocellulose membrane, and then we seal it in the grid with the solid support matrix. That is, we have a sponges on the both the side, and then we have a grid kind of a technique, grid supporting grid, and then we keep it under a chamber. Under the chamber, we have a buffer, and we run under the current. So this time, whatever DNA is there on the gel will travel to the membrane, that is nitrocellular membrane. So exact replica of the gel, the bands which are formed in the gel will come on the membrane. And now what happens? The next we go to the probing technique, or also called as an hybridization technique. Now let's say I want a particular DNA sequence, and I want to know whether this DNA sequence is present in the given sample or extracted sample or separated sample. I just use the sequences, label it with the radio label, and then we give it to the, we add this probing, radio label probes to be interacting with the nitrocellular membrane DNA, and whichever DNA is able to interact, they will be having an annealing, and then the hybridization we say, and we observe under the X-ray. So at this point of time, radio labeled annealed DNA will have the color formation. So that is a visualizing fluorescence kind of a visualization they are able to give it. So in the southern blotting, we are using the DNA for the detection. Later we'll go to the northern blotting. Northern blotting goes similar like southern blotting. In the northern blotting, we have sample. We extract the RNA. The RNA extracted, we run in the electrophoresis chamber. Electrophoresis again, we can use agarose gel electrophoresis for northern blotting. After that, we are transferring this gel to an nitrocellulose membrane, and this membrane we are supporting in the chamber. We are adding it into the grid system and then keeping it in the buffer. Uh, Once we keep it into the buffer, what happens is through the capillary action, the current will also be running overnight. Running, we are doing it using the buffer. Now this, whatever the replica of the DNA is there, RNA is there, that will get transferred to the nylon membrane. Now this nylon membrane now again taken and used for the probing technique or hybridization technique. Here this membrane is incubated with the targeted RNA sequences, and then we use under detection using the X-ray or any other technique. So this is how exactly northern blotting is carried out. Then we have western blotting. Unlike the RNA and DNA, electrophoresis occurs through the agarose gel electrophoresis, but the protein separation happens to the page or STS page, we say polyacrylamide gel electrophoresis. You can look at the other videos to understand the polyacrylamide gel electrophoresis. And here protein gets separated based on their size and charge. Once they are separated, again we are transferring it to the membrane, so the nitrocellulose membrane, and then we have a chamber where we keep the grids and make it easy for the transfer of the protein. So once the protein is transferred, the nitrocellulose membrane is now. So here, unlike the probing the radio label technique, in this technique we use the method of the ELISA. What exactly we are going to do is we are going to keep this nitrocellulose membrane same. We are using antibodies here. We will be adding antibodies because we know the proteins will not interact with the proteins binding. So it will be difficult to understand in the annealing sequences, which is not possible with the proteins. So here we will add the primary antibodies, which is going to bind specific to the certain proteins, and then we add the wash it and then add the secondary antibodies. The secondary antibodies will be uh, enzyme linkers. So we have spoke about this in the enzyme link, and then we see under the when we add the appropriate substrate along with the buffer, it just gives the visibility. So basically, in this technique, Western blotting technique, we are not using the protein as a probe here, but we use the antibody interaction with the protein. Where exactly we use two types of antibody. The primary antibody will be binding to the protein and followed by the secondary antibody, which is conjugated with the enzyme, bind to the primary antibody. And this enzymes, later on adding the substrate, gives the color. So this is how exactly the Western blotting technique is used. So these are the techniques which is used for the separation or the detection of the RNA, DNA or protein. So what are the membrane we can use? You can see here we have nitrocellulose membranes. We have nitrocellulose membranes are used for the DNA and RNA blotting technique. We have PVTF membranes which is used for the protein blotting technique. Also we have the nylon membranes which also can be used for the various DNA, RNA as well as the protein blotting.